much everyone here. So I'm going to start with just a very brief introduction of myself and my career path up till now, and the type of work I do and why I do it. And then I'm going to jump into some specific examples of my studies and hopefully provide a little bit of something for everyone, but it's going to be like a bit of a whirlwind tour. So just brief housekeeping, I have no relevant disclosures related to any of the work that I'll be discussing today, but in fact, here's a list of my currently funded research portfolio, and I've highlighted in yellow the ones that will get some airtime today, basically my two R1s and my two APS research scholar grants to some extent. And you can see that all these projects are either federally funded or funded by foundations or internally funded. So, by way of background for me, I graduated in 2003 with my undergraduate degree from the University of Virginia, and at least two people in this room will recognize the person sitting next to me in that picture, who at the time was my boyfriend and now is my husband, and the other woman in the picture was my best friend, and still is my best friend, so at least that implies consistency of character in my heart. So nothing else, you can take that away from today's talk. And then in 2011, I graduated with my PhD from UNC in health policy and management. And that's my thesis advisor, Morris Weinberger, just retired last year, which makes me feel like I'm getting old. And then up until through December 2020, I was faculty at Duke. This is me right here. This is a stock photo. It has nothing to do with anything that I ever did at Duke. I just think it's hilarious. And so I like to use it as my official photo. And then starting January 1, 2021, at the height of the pandemic, I joined the faculty at Yale. And so for the first 2 years of my job at Yale, this is basically what my work setup looked like, including my coworker here, who just likes to randomly delete emails. And then obviously over time, we've been become more in person again. So for organization's sake, I can bucket my research projects into 3 broad categories. And so I can say emerging technology and oncology care, survivorship and patient outcomes, and then molecular oncology outcomes research. But the running theme throughout is this leveraging the strengths of different data sources to study emerging technologies in cancer care and how these advances impact health disparities, outcomes, and costs. So not broad at all. And I think I can safely assume that everyone here is very familiar with this level of evidence pyramid and the fact that according to this dogma here, that RCTs are considered higher up the food chain than cohort and case control studies. And as researchers in cancer prevention and control, I also assume that many of you spend a fair amount of time thinking about how there are many types of questions that are not feasible to examine in the context of a trial, but that are feasible with other health outcomes study methodologies. And so I really prefer to think about these different study methodologies as complementary, where you can ask and answer different types of questions with the common goal of improving cancer care. And I hope that comes across in my talk today. And these are just some of examples of the more common types of questions that we can answer about emerging cancer diagnostics and therapeutics using real world data sources. And you will see that many of these themes will show up over and over again in the work that I'm going to talk to you about today. So sometimes we're thinking about putting in grants or doing research projects. It can seem a little bit like a black box, but I think there's actually some kind of like framework that we can provide around thinking through these different types of questions. But I always like to start off here, and this is a reminder that we know that patients who participate in RCTs differ systematically from the average real-world patient, where life and treatment is just a lot messier as compared to the highly curated patient population and the controlled environment of an RCT. And this is an example of studies of patients with primary CNS lymphoma treated at the same institution who received the same treatment, just on versus off protocol. And they showed patients who were treated in real world practice, meaning off protocol, were older, they were sicker, they had worse disease, and had dramatically worse survival than patients who were treated on the clinical trial. And I think it's important to note that this study does not reflect differences in access, since all of these patients were being treated at the same institution with the same treatment, just on versus off protocol, although we know that access is an additional huge concern in the real world setting, and is something that I'll talk a little bit more about in the rest of my presentation. So here I have presented an overview of many of the different types of data that can be used to conduct real-world health outcomes research in oncology. And I often say that this slide most accurately represents what the inside of my brain looks like as I'm considering a new research project and trying to figure out the best way about going about answering a specific clinical question of interest, which is probably the topic I get asked to talk about the most. What kinds of questions can we answer using data that are currently and readily available? And this is just a particularly maybe salient point from people who are more junior in their careers, but relevant to everyone. I'm a big believer in that it's really important to remember that there's no perfect single data set. You're never going to get a data set that's going to be perfect to answer every aspect of every question that you're interested in. But I've built a career off of leveraging the major strengths and kind of navigating the central weaknesses of these different data sets as they currently exist, and then also considering novel data linkages. And by doing these things, we can answer some pretty cool questions. And I hope we convince you of that today. So I'm going to start just talking about a study from one of my current R01s, where we were examining access and adherence to oral anti-cancer agents, and specifically looking at real-world disparities in patients with metastatic renal cell carcinoma. And as is the case with many cancers, the number of available therapies for kidney cancer have expanded dramatically over the past 15 plus years. And interestingly, of the 10 therapies approved between 2005 and 2016 for renal cell carcinoma, 7 of them were oral agents. And again, things have, even since the start of this grant, changed dramatically again, and we'll get to that. But the idea was that we could use real-world data to look at issues pertaining to patients' ability to both access and then stay adherent to these oral therapies. And this was actually in response to a specific notice of special interest that was specific to oral therapies. So I always like to start with thinking about what do we currently know versus the knowledge gaps. So we're thinking about OAA use in kidney cancer patients. So we know that oral agents pose unique challenges to delivery, right? So oral therapies, very obviously, but something we don't necessarily think a lot about. Patients take them on their own. They go to a pharmacy, they fill the prescription, they go home, they're all kind of on their own. Whereas for like traditional kind of chemotherapeutic agents, they're administered in a clinical setting under the supervision of healthcare providers, right? And that we also know that clinical trial data had shown that there was both increased progression-free survival as well as overall survival for several of these OAAs. And they were generally thought to have a pretty favorable toxicity profile as compared to traditional cytotoxic chemotherapeutic agents. But we also knew that there were significant knowledge gaps around the use of these types of therapies, specifically looking at outcomes and toxicity in older patients and patients with more comorbidities who were not included in clinical trials to begin with. And that there were unknown barriers to patients accessing and then staying adherent to these drugs, some of which would include things like cost, which had not been examined before. And then we didn't have a great sense of what non-adherence looked like, or how non-adherence played a role in outcomes for these patients in the real world. So again, just taking you through kind of our thought process and putting this study together, we decided to leverage the strengths of the SEER Medicare and the Medicare claims data alone as well. And in this case, the inclusion of Medicare Part D was obviously critical because we needed the prescription drug claims. And then we also used an additional data source called the North Carolina Cipher data. And I don't know how familiar people are with this data set, but I'll just give you a basic overview. So North Carolina Cipher is North Carolina's cancer registry that has been linked to Medicare, Medicaid, and Blue Cross Blue Shield claims data. But the important aspect to consider in the context of what we were trying to do here is to think about its strengths and limitations in comparison to the other data set that we were using. So the strengths of the North Carolina Cipher data includes the same detailed clinical pathologic information that's available in the SEER Medicare data. In fact, it's structured very similarly because they both use the NACER format. But it allows us to look at patients of all ages, so we're not limited to those who are 65 years and older, and it also allows us to look at different types of insurance coverage as well. So here, again, just starting with utilization, I show the SEER Medicare rates of utilization We also did reproduce these analyses in the North Carolina Cipher data, where we saw highly similar trajectories and rates of all of these different oral therapy agents. And we found like roughly a third of patients were receiving an oral anti-cancer agent within a year of being diagnosed with advanced disease, and that the majority of these patients were initially treated with the drug sunitinib. So when we moved to multivariable analysis of serum Medicare factors associated with utilization, we actually did not show evidence of differential receipt of oral therapies by patient race or ethnicity or socioeconomic status in these initial analyses, but we did see decreased utilization in patients who were unmarried, older, or that lived in the South. And again, these are things that we've kind of seen consistently in other studies in other spaces. So one of the added strengths of the North Carolina Cipher data is that it includes adults of all ages as well as private insurance. And so we looked at these same questions within the North Carolina Cipher data as well. And what we saw was that before we adjusted for age, there were large differences in utilization by private versus Medicare insurance. However, in multivariable adjusted analyses, we saw that there was no difference in OA utilization by insurance status. And instead, we found that this was likely driven entirely by age, with older patients being less likely to receive therapy. And we also observed that frailty and having multiple comorbidities were both associated with decreased OA utilization as well in this population. And then lastly, in the Cipher data, we looked at patients with all stages of kidney cancer and saw that patients who were diagnosed with stage 1 disease, but then experienced progression to metastatic disease, were less likely to utilize an OAA within a year of metastatic disease diagnosis. And we think that this is likely due to slower growing disease with a less urgent need to treat immediately. So for oral anti-cancer agents, we talked about utilization, but it's also important to remember that in addition to utilization, there's this concept of adherence, or the percentage of time a patient was taking their anti-cancer drug. So we know that in general, adherence to oral medications is often far from 100% due to any number of reasons that can include things like side effects or costs. And so we were interested in looking at adherence separately because we thought there might be different factors that are impacting a patient's ability to initially access a drug versus a patient's ability to then stay adherent to that drug. So we looked at adherence in both the SEER, Medicare, and the Cipher cohorts. So we looked at them separately, but we can kind of look at the similarities that we observed in both. And we observed slightly higher rates of adherence within the North Carolina Cipher patient population as compared with the SEER Medicare cohort. And again, we suspect that's because of the inclusion of the younger patient population. And then both cohorts showed evidence of either older patients or those with Medicare insurance having lower adherence rates. So the North Carolina Cipher data was somewhat limited in power due to smaller sample sizes. So we were not able to examine adherence by agent in multivariable analyses. However, there was a substantial evidence of lower adherence to sorafenib in both cohorts. And we saw a strong impact of poverty on adherence within the SEER Medicare data, but not within North Carolina Cipher. And then, although it's unclear why this was the case, we hypothesized that older patients living on a fixed income may be more sensitive to financial stressors. And then we found that consistent with this hypothesis, that OAAs with out-of-pocket costs over $200 were associated with decreased adherence in the SEER Medicare cohort. And we could not look at costs in the North Carolina Cipher data because costs were not available in that data set. So we can also use these data sets to look at survival. And so this is a 3-month landmark survival curve of cancer-specific mortality for pazopanib users by whether they received the trial recommended single dose of 800 milligrams of pazopanib per day in the 3 months following their OAA initiation. So for the patients who were getting the prescribed dose for the first 3 months of treatment, we saw superior outcomes, and survival was assessed at beginning at 3 months post pazopanib initiation in order to avoid introducing immortal time bias. So as a reminder, the third data set that we're leveraging in this study is the Medicare 100% data. And we're doing this so that we can examine one, nationwide utilization, because the SEER Medicare 100% data is one of the few ways that you can actually look at a nationally representative data source. And then two, so that we can also use more recent data to look at oral therapies in the context of newer emerging therapies such as immuno-oncology agents. So when we initially submitted this study, like I said, it was in response to a NOCI specific to oral anti-cancer agents, but we knew like that the landscape of treatment for patients with metastatic renal cell carcinoma was in an upheaval, and so we knew that we wanted to have the ability to look at the context of these oral therapies in the context of immunotherapy, but we also knew that the SEER Medicare data was going to lag too significantly for us to really be able to look at that with any sort of nuance. So that's why we went in for the Medicare 100% data as well, even though knowing that would introduce a limitation where we wouldn't have the specific stage information. But in our Medicare 100% analysis, we saw that the proportion of patients receiving any systemic therapy increased from about 51% in 2015 to 59% in 2019. And again, this is the time period in which immunotherapies are really taking off for the metastatic renal cell carcinoma patient population. And we also saw that the use of IO as initial therapy increased from around 4% in 2015 to 37% in 2019. And while OAA use decreased from 31% in 2015 to about 11% in 2019 as initial therapy. And we see that IOs first surpassed OAAs as initial therapy in 2018. Again, this landscape continues to change because now combination therapy is kind of moving to the forefront of what people are looking at, but very quickly changing landscape of treatment for these patients. We also examined the use of IO or OAA treatment by race, and we saw that non-Hispanic Black patients received IOs at lower rates than non-Hispanic white patients, and they also received OAAs at lower rates than patients of other race and ethnicities. So due to the small sample sizes, multiple races and ethnicities were combined for visualization purposes here, as indicated in the figure, which is not ideal, but we need to do that for our small sample size issues. And these data are expressed as unadjusted percentages of patients diagnosed with metastatic renal cell in a given year. But these findings persisted in multivariable analysis where we saw that non-Hispanic Black patients had both lower IO and OAA received as well. And so I think it's really critical to acknowledge that a key limitation of all these data sets is the lack of the patient perspective and the patient voice. So this is missing from the secondary data analysis. And I also feel like it's incredibly important to do our best to include this perspective, even when working exclusively with secondary data. And so one way that we tried to address this was by partnering with patient advocacy organizations and patients themselves throughout the duration of our entire study. So before we even touched data, we met with our patient advocacy organizations and our patient representatives, and we presented to them our proposed analysis and just said, hey, like, this is what we're planning on doing. This is what we think the interesting questions are. What are we getting right? What are we missing? And one really interesting thing that came from this very first study that we had was that we heard from patients directly that they were very concerned about how often their oral medications were being switched. So from their perspective, they thought that perhaps their providers were too quick to switch them off of an oral therapy when they experience a toxicity. And they would have preferred to stay on that oral therapy for longer, trying to manage that toxicity rather than immediately switching. From their perspective, it felt like they would be potentially missing out on the life-saving benefit of that, the potential life-saving benefit of that drug if they were switched too quickly. And then that would kind of like burn through their options more quickly, so to speak. So they asked if that was something that we could examine in our data set, and we could. And so we added this for analysis, and we're able to show that within 90 days of diagnosis, we saw about 6% of patients were switched in the therapy. And then when you extend that out to 1 year, about 20% of RCC patients are switched to a different oral therapy. But this was very important to them. And so this brings me to a brand new R01 that we have. And so I don't really have any data to present on this. I just want to show you kind of like the similar ways that we've gone about thinking about this. And again, you're going to recognize these similar themes. But this is a R01 looking at broad genomic profiling in patients with advanced lung cancer. And I am NPI on this study with Carrie Gross, who's also at Yale. And we have this fantastic team of collaborators, but you'll just notice some similarities in the way that we've structured these aims. So we use a lot of the same kind of ways of structuring this data set. So for aim or structuring this study. So for AIM-1, we're looking at utilization of broad genomic profiling across actually multiple different cancer types. And then in AIM-2, we're specifically interested in looking at the results of these broad genomic profiling tests and their impact on clinical decision-making in advanced non-small cell lung cancer. So we're going to move into the lung cancer space. And then AIM-3, we want to look at questions of how BGP is associated with costs and survival. And then we're going to bring it all together for AIM-4 and a cost effectiveness and value of information analysis. And so I just wanted to give you a sense that how we once again are just leveraging the strength of multiple data sets to answer different aspects of a broader clinical question of interest, because we can't answer all these questions in one data set. So for the one for AIM-1, where we're interested in looking at kind of utilization of these tests across multiple different cancer types, we're going to be looking in the Medicare fee-for-service and Medicare Advantage data sets, as well as the Blue Cross Blue Shield data, to get kind of a bigger picture look at what the utilization of these tests look like, how they've changed over time, those types of questions. And then for AIM-2, where we want to look at how the test results have impacted treatment decisions, we need to move into data sets that have test result information. So we're going to be looking at the CancerLinQ data, which is now the ConcertAI data, and the Flatiron Health data, both of which have different types of test result information available. And again, once we move to the AIM-2, we're going to look at looking at non-small cell lung cancer specifically. And then for AIM-3, we're going to move into the Serum Medicare data, because again, we want to look at questions of costs and survival, and those have the best kind of data in that space. And then this is just the overarching conceptual framework for the study, which shows how we would use these different retrospective data and complementary data sources to investigate the adoption and clinical impact of BGP in real-world settings, with the resulting evidence then being incorporated into this cost-effectiveness value of information modeling, where we can assess things about the strength of evidence or the magnitude of the benefit of these tests. And this figure just shows the steps in a value of information analysis. And our study really acknowledges that the value of BGP in cancer is a moving target. It differs across tumor types. It's evolved with the changes in drug development and the changing in underlying clinical evidence, and it's hindered by gaps in test results and receipt of recommended care. So value of information approach can provide an empirical approach to guide decision making and can really help inform questions like, do we need further research? And what evidence should we be collecting, and how should we design and prioritize further research going forward? So now I'm going to move on to another example of my current work, doing something a little different. But again, you're going to see the same kind of themes, but we're doing something quite different in this study. And this study is funded by an American Cancer Society 5-year research scholar grant. And the purpose of this work is to develop and validate a survivorship risk model to inform pathways of care for long-term cancer survivors. So the second stage for this study, and we know that due to advances in early detection and treatment and oncologic outcomes, many cancer survivors are now living longer and are thus more likely to die from and or develop conditions other than their original cancer. And in fact, 2/3 of cancer patients now live past 5 years. And this is a figure that's modified from a paper by Effinger and McCabe, and shows at the top the current model of care for cancer survivors, which is more of a one-size-fits-all approach, where once a patient is diagnosed with cancer, their care is transferred to an oncologist for an indefinite period of time, with little to no ongoing participation from primary care. And the bottom shows a proposed shared practice model of care based on risk stratification, which helps to inform the point in time when a cancer survivor's care might be appropriately transferred back or shared with primary care physicians, or bringing on other members of the clinical care team that are necessary to their long-term care, with the idea being that this new model represents both a more efficient and a better quality model of care. And this figure is from a study where McConnell and colleagues used National Cancer Registry data from the UK and Northern Ireland to risk stratify patients with 20 of the most common cancers into 3 groups based on overall survival at 1 and 5 years from diagnosis. And this is just to demonstrate that crude risk categorization is both possible and is currently being used to inform care in other countries. This is already being done in other countries. And the authors of this study noted that the important caveats of this analysis included an absence of treatment information, which was not available in their cancer registry data, and that their data were unable to assess treatment-related complications because there was no treatment-related data. And both of these we can improve upon in our risk models for this ACS grant. So again, this is already being done at a crude level elsewhere. So given this background, we know that oncologic and non-oncologic risks vary substantially by cancer stage, or cancer type, cancer stage, and treatment received. We also know that cancer site and stage alone can provide broad oncologic risk categories. It's already being done elsewhere. But we also know that non-oncologic disease risks for cancer survivors have been qualitatively but not quantitatively well defined in cancer survivors. And how oncologic and non-oncologic risks compare with within the cancer survivors remains an unknown question. And the idea is that how can we estimate these risks at the point of care? So this current study aims to provide robust risk estimates of oncologic and non-oncologic needs in cancer survivors available for managing physicians at the point of care. And for the sake of clarity, we define oncologic risks as those related to recurrence or progression of a patient's primary treated cancer. And these events would typically be best managed by a patient's primary oncologist. In contrast, we define that non-oncologic risks as all other health issues for which survivors are at potentially increased risks, typically due to either the physical or psychological sequelae of their cancer treatment. So for this overall ACS proposal, we are looking at breast, prostate, colon, and rectal cancers separately. We're modeling them all separately. And we began by thinking about what detailed information about staging and treatment would be available for each of these cancers across different data sets, because we knew we wanted to develop these risk stratification models, and we knew we wanted to validate them. So we would need consistent information across multiple different data sets. And we settled on 4 different data sets with different strengths and limitations, which we'll use over the course of the study. And the overarching plan of the study is to use inputs available from all of these data sets, such as cancer-specific variables, such as site, stage, and treatment, personal characteristics like age, gender, race and ethnicity, marital status, and then aging-related concerns such as comorbidities and functional status. And we're going to use these to develop risk prediction models, again, in breast, prostate, and colon and rectal cancers, to predict both oncologic and non-oncologic outcomes, events for which long-term survivors are at increased risk. So our oncologic events will include death from their primary cancer, as well as the progression of their primary cancer, and the non-oncologic risk will include a new CVD event or death, the development of a subsequent, new primary malignancy, development of frailty, and then a major depression event. So we identified these as our starting place. I'm sure there are more, but this is where we're starting. And these risk algorithms will separate long-term cancer survivors into low, medium, and high-risk categories to help inform discussions between survivors and physicians about their optimal care going forward. I'm just looking at the time. Okay. And ultimately, the final product is going to be a freely available web calculator in which patients and/or physicians, maybe together ideally, can input their individual information just to help categorize their individual risk and inform pathways of care. We are not dictating what those pathways of care are. We are informing or providing additional information to help inform what those decision points should be. So we're still early in the process, but the study will use the SEER-only data as well as the SEER Medicare data. SEER will be used for the first phase of risk prediction, and it'll be a non-claims-based approach. But then we'll use SEER Medicare for the claims-based prediction. And then obviously, once we do that, that's going to limit our analysis to those who are 65 years and older. But we are then going to independently validate our algorithms in the North Carolina Cipher data, which I've already talked to you about a little bit, and then we will further validate it in a VA cohort from their VA cancer registry data, which also has similar data elements in it as compared to SEER Medicare and the North Carolina Cipher data. So now I'm going to present the results from our SEER-only analysis, in which we examine the relative burden of oncologic and non-oncologic mortality in long-term cancer survivors, and determine the absolute and relative risks of cancer-specific versus non-cancer-specific mortality among long-term survivors across these 4 different cancers. And it's important to keep in mind, making sure you can see that, yeah, that this study is focusing on long-term cancer survivors, which are we're defining as those individuals who have survived at least 5 years past their initial diagnosis. And this 5-year benchmark is somewhat debatable, it's somewhat arbitrary, but it remains a practical window after which non-oncologic follow-up is often considered for many cancers. And then, for example, the National Institute of Health has used a 5-year horizon in multiple requests for applications to investigate long-term cancer survivors. So you could argue, is 5 years the correct answer? Maybe, maybe not, but it's at least accepted as a benchmark for long-term cancer survivors. And so here's our consort diagram for this study showing that patients with breast, prostate, or colorectal cancer diagnosed between 2003 and 2014 in the SEER data. And then our exclusion criteria included kind of the common exclusion criteria that you often see in these SEER analyses, where we were excluding people who were diagnosed with autopsy, people who were diagnosed with multiple cancers, and people who died within 5 years of their diagnosis. And then this got us to a final cohort that included just over 350,000 breast cancer patients, just under 120,000 prostate cancer patients, around 100,000 colon cancer patients, and just over 40,000 rectal cancer patients. So once again, our outcomes of interest were cancer-specific mortality versus non-cancer-specific mortality. So mortality due to the primary cancer was classified as cancer-specific mortality, pretty straightforward. Mortality due to causes other than the primary cancer was classified as non-cancer specific mortality. So for a patient whose index cancer was colon cancer, a death from any other cancer, as well as any non-cancer condition, would be classified as a non-cancer specific mortality. All demographic and clinical factors were collected at the time of diagnosis, including age, sex, race, and ethnicity, year of diagnosis, income and urban versus rural status. And then we also looked at tumor characteristics such as stage, grade, and nodal status. So we used lasso regression to select the factors associated with cancer-specific and non-cancer specific mortality separately. We used lasso because we wanted to create parsimonious models. We were not looking to do the AI version of this. Variables with a regression coefficient equal to 0 after the shrinking process were excluded from the model, and variables with non-zero coefficients were included in the survival analysis. So the proportional hazards assumption was not satisfied for several of our covariates. So for our survival analysis, we used accelerated failure time models, or AFT models, which do not rely on the proportional hazards assumption. And then the parameter estimates for the AFT models were converted to time ratio estimates to interpret the effect of a covariance on a time scale. But the important thing to remember is that a time ratio less than 1 indicates a shorter survival or an unfavorable outcome. And then patients were grouped into 3 risk groups of oncologic mortality: low, intermediate, and high. And I'll talk a little more specifically about that in a second for each of the different cancers. And then cumulative incidence function curves for cancer-specific and non-cancer-specific mortality by risk group were generated for all cancer types. And so just quickly, in terms of descriptive statistics, what we saw in the overall cohort was that many of the patients were over the age of 65, even though we were looking in SEER. So our breast cancer cohort was our youngest cohort with 34% over the age of 65. Our colon cancer cohort was our oldest cohort with 54% of patients over the age of 65. Across all cancer cohorts, more than 70% of patients fell into the non-Hispanic white category. And then greater than 30% of patients across all cohorts were diagnosed with stage 3 disease. We looked at non-cancer causes of death across the cohort, and we saw that more than 2/3 of these patients died of non-cancer related causes. And of these, about a third were attributed to heart disease. And then other common causes of death were COPD, cerebrovascular disease, and Alzheimer's. Yes.
Just a quick question. So is it long-term survivors? Does that mean they all had evidence of disease at that 5-year mark?
So everyone was diagnosed, and then they had to survive 5 years.
5 years.
And then we looked after that point.
So they might have.
They may or may not have had evidence.
Oh, okay, thank you.
Yeah, but they had to have survived to that 5-year time point, and then we started looking for.
Thank you.
Okay, so we're going to start with the results from our breast cancer cohort. So for our survival models, we saw that for breast cancer patients, high stage and grade were associated with decreased breast cancer specific survival, whereas ER status was associated, ER negative status was associated with increased breast cancer specific survival. And although the ER negative disease having a more favorable survival might be surprising at first glance, again, I will remind you that we are looking at the survival of patients who've already survived 5 years out from their initial diagnosis. And having ER negative disease is therefore actually favorable in long-term survivors, since late relapses are relatively uncommon compared with ER positive disease. So it actually makes sense in this context. And then for breast cancer specific mortality, for non-breast cancer specific mortality, on the bottom, we saw that higher age and stage were associated with decreased survival time. So as I mentioned before, the next step was that we grouped patients into 3 risk groups of oncologic mortality. For the breast cancer patients, these groups were defined as, for low risk, it was patients defined at older than 65 years of age and stage 1 disease. And then for the high risk category, it was patients diagnosed at less than 65 years of age with stage 2 to 3 disease. And then that intermediate category included everyone else. And then we looked at cumulative incidence function curves for cancer specific and non-cancer specific mortality by these different risk groups. So in looking at the cumulative incidence of non-cancer specific mortality, so on the left, this is for the low risk group. Yeah, the left. The cumulative incidence of non-cancer specific mortality in orange in the low risk breast cancer group was 6.7 times higher than the cancer specific mortality in the blue. When you travel over to the right in the high risk group, you can see that those lines flip, and that the cumulative incidence of cancer specific mortality in the blue is actually 2.4 times higher than non-cancer specific mortality in the orange in the high risk breast cancer group. So let's look at how this compares to what we see in our prostate cancer cohort, because again, we modeled all of these separately. And when we look at our survival models, we can see that higher age, PSA, and Gleason score were all associated with decreased survival time for both prostate cancer specific mortality as well as non-prostate cancer specific mortality. And we think that this is because later stage of diagnosis or worse disease associated with different treatments may impact downstream sequelae for these patients. We try to be pretty consistent with our categories. Obviously, we have to make some changes depending on the cancer type. But again, you can see for our low risk category, we included patients diagnosed over the age of 65 with Gleason 6, high risk less than 65 with Gleason 7+, and everyone else again in the middle. But when you look at our cumulative incidence function curves for cancer specific and non-cancer specific mortality by risk groups for prostate cancer, we see something different than we saw in breast cancer. So again, on the left, when you're looking at the low risk group, we can see that the cumulative incidence of non-cancer specific mortality in the orange in low risk prostate cancer patients is 8.6 times higher than the cancer specific mortality. And when you travel over to the right for the high risk group, you can see that they don't flip, and that the cumulative incidence of non-cancer specific mortality in the orange in high risk prostate cancer patients is still 1.8 times higher than the cancer specific mortality in blue. So I'll go ahead and spoil it and tell you that's the only place where we saw that happen was in prostate cancer. In colon and rectal cancers, it was much more consistent, different effect sizes, but much more consistent with what we saw in the breast cancer cohort. So with patients with stage 3 colon cancer compared to stage 1, they had a 40% reduction in median survival time for colon cancer specific mortality, and an 8% reduction in median survival time for colon cancer specific mortality. And this is compared to stage 1 rectal cancer patients, comparing stage 1 rectal cancer patients with stage 3 disease had a 29% reduction in median survival time for rectal cancer specific mortality. However, there was no association between stage and non-rectal cancer specific mortality in the non-rectal cancer specific mortality group. And then again, our risk categories were the same for colorectal cancer patients. And then when we looked at the cumulative incidence curves for colon cancer, again, you can see the non-cancer specific mortality in orange. In the low-risk colon cancer patients, it's 7 times higher than the cancer-specific mortality. You see that flip happen again, maybe not to the same extent in the high-risk group, where the cancer-specific mortality is 1.5 times higher in the high-risk group than the non-cancer specific mortality. And then for the rectal cancer group, we saw that flip happen again. So our main takeaway points from this study, and this study was published in JAMA Network Open just about a half a year ago, are that patients with low oncologic risks have higher mortality related to causes other than their initial cancer across the board, and that heart disease, Alzheimer's, COPD, and cerebrovascular disease were the dominant causes of death among patients with low oncologic risks. The limitations of this study is that there was no internal or external validation, and there was in this case, we only used the SEER data, so we didn't have information on treatment, or we had limited data on treatment and progression. And then the coronavirus data were not available. So where are we going next with this? So we've already moved into our SEER Medicare analysis, which allows us to look and build these models for these other outcomes, because this gives us the information we need to assess more detailed treatment information, as well as some of these different outcomes of interest. So we're looking at subsequent malignancy, frailty, cardiovascular disease, and depression. And then we're doing both an internal validation within the SEER Medicare data set, doing a training and validation set, separating our cohorts into training and validation sets, as well as an external validation we'll be conducting in the North Carolina site or in VA data sets. So again, I'm gonna beat this drum of it's important to have like diverse kind of voices in your project. So we have an external advisory board. It includes both giants in the field of cancer survivorship, but also includes cancer survivors themselves, to make sure that we're hearing their voices, and that when we're talking and thinking about these questions, that it's representative of their concerns, and that when we're disseminating these findings, it also includes their voice. So the very last thing that I'm going to talk about, and this is going to be very brief, is a little bit of a sneak peek of what I think the future, or one part of the future of health outcomes research in oncology will look like. And this starts with a novel pilot project that we conducted, where we created this novel linkage in a breast cancer cohort of SEER, Medicare, and residual tissue repository data, so physical tumor specimens for these patients. And the purpose behind this novel linkage was to tackle additional unmet needs. I've talked a lot about leveraging different strengths and weaknesses of data sets, but we wanted to tackle additional unmet needs of traditional health outcomes research by developing studies that will include actual physical tumor samples, so that we can run genomic sequence analyses on them, kind of whatever, the world's your oyster, and then link that additional biologic information to both tumor registry and longitudinal claims data to get an even more complete kind of idea of what's going on for some of these clinical questions of interest. So there's a couple of existing resources which our team has already tapped into to get this work off the ground. And one of these that we used for the pilot study was the SEER Residual Tissue Repository, which is a program that was funded by NCI to maintain physical tumor samples for patients included in the SEER registries for 3 participating sites, Iowa, Hawaii, and Los Angeles. And when it was funded, they funded pathologic specimens collected for these registries and banked between 1992 and 2006. Some of the registries continue to do that process now. And this allows for the ability to link the SEER Medicare data to the physical available tumor samples. And then we have worked with NCI to then do a novel linkage with the bringing in the Medicare claims piece as well. So a few years ago now, we completed this proof of concept study on a very small breast cancer cohort to demonstrate the process for combining the serum Medicare and the genomic or biologic data obtained from running gene expression analyses on the tumor sample. So for our pilot study, unfortunately, LA did not participate due to the inability to procure large enough funds to cover their participation costs, but we did work with Iowa and Hawaii, who were fantastic to work with. But this did leave us with two very distinct and racially and ethnically homogenous populations, which was not ideal, but we were still allowed us to proceed with our proof of concept study. And this is just a brief overview of some of our major findings. And again, this study was published in JAMA Network, and I'm happy to share the link for anyone who wants to go to this in more detail. But you can see the different pieces that we were able to pull from these different data sets, right? So like using the Medicare claims data, we could use a claims-based approach to look at symptomatic versus non-symptomatic detection of disease. And we saw, again, we know this, but we saw that symptomatic detection of disease was associated with increased mortality. Using the SEER registry portions of the data allowed us to look at things like zip code, high school graduation rates. And again, not surprising, but we saw that low zip code high school graduation rates were also associated with increased mortality. But then by adding the tumor samples and the genetic analyses that were conducted on these, we also saw things like androgen receptor macrophage cytotoxicity and Treg signaling were associated with reduced mortality. And when we combined all of this information into one model to look at outcomes for these patients, we saw that factors related to socioeconomic status and screening access remained associated with mortality even after adjusting for all clinical and genomic factors. So we were the first to be able to demonstrate that. So this is a brand new study that is going to build off of that just got funded as of February 1st of this year. And we're going to look at disparities in the molecular etiology of breast cancer. And so the approach here is we really want to compare this patients. All these patients will have screen diagnosed cancer. And we want to compare patients who are diagnosed with favorable versus unfavorable screen detected breast cancer, and then infer the molecular etiology through something called mutational deconvolution analysis. And our goal is really to assess whether the molecular etiology varies by favorable versus non-favorable disease. And then we're also going to look at this in the context of different socioeconomic factors. But really, we know that breast cancer mortality has been successfully reduced with the widespread adoption of screening mammography. But we also know that a subset of women continue to be diagnosed with aggressive cancers, even though they're undergoing screening, and that these have become the dominant driver of cancer mortality. And the ability to directly study the underlying etiologies driving aggressive breast cancers has not been previously possible for two reasons. One is the lack of population level data linked to physical tumor specimens, which allows the conduct of the necessary genomic analyses. And two is a lack of the bioinformatic methodologies that are capable of inferring causality at the individual tumor level. So our study is really focusing on the underlying etiologies, differentiating what are often called like the tortoises and the hares of breast cancers versus the late stage or birds of breast cancers, which continues to pose a disproportionate risk of mortality to women with screening detected breast cancer. So again, everyone's getting screening, it's just whether they're diagnosed with favorable versus aggressive disease. And for our study purposes, we're looking at screening detected disease, ER, PR positive, HER2 negative, but then categorizing, then separating patients into these favorable and aggressive categories based on stage and grade. And then we can pull these different pieces of information from these different aspects of the data set. So we're partnering for this study, we're partnering specifically with the Connecticut SEER registry, which is again going to allow us information about diagnosis, stage, and survival. We're linking it with the Medicare claims data, which will give us information about the, allow us to kind of like cherry pick the cases for women with screen detected breast cancer. And then we're going to separate our cohort into patients who have this kind of more aggressive versus more favorable phenotype. And then using an approach developed by our colleague and co-I, Jeffrey Townsend, who's also at Yale, where we will infer molecular etiology via mutational deconvolution analysis, where we can examine the contribution of different mutagens to the driver mutations found within each patient using whole exome sequencing. And so Dr. Townsend has previously demonstrated this technique, and the reference there is at the bottom. It's a very interesting read. And he's previously demonstrated this technique across multiple different cancers. And an example is that, like in skin cancers, he was able to show that characteristic thymidine dimer mutations caused by ultraviolet light are largely responsible for the mutations that drive the activity of skin cancers. But we're basically able to look at these different mutagens, some of which are both preventable, some of which are non-modifiable, and see if any of those are predominantly showing up as the mutational fingerprint in these two different categories of disease. And that's what we will be doing, and we're very excited about it. So that's it. There are many questions relevant to cancer care that can be informed by real-world health outcomes methodologies. There are several data sets available for real-world cancer outcomes research, and there are many questions that can't be feasibly or ethically answered in a trial setting, but we can leverage the strengths and weaknesses of different data sets to kind of answer some of these questions, and then novel linkages may pave the way for different opportunities, and the incorporation of genomics and biology into health outcome research is here. All my science is team-based science. I cannot, working with these teams elevates what any one of us could do on our own. I think that last example of our newly funded study is a perfect example of that. Like it literally couldn't be done by one of us. It's taken all of us together with our own individual expertise to accomplish that work. And so these are the large teams of biostatisticians, clinicians, all across different specialties, basic scientists who are working with me to achieve all of this work. And that's it. Sorry, we're like almost at time, but I'm happy to take questions. Yeah.
So with your community advisory group that you work with, what's been like one of the more recent questions that you found is like engaged or re-engaged that group like the strongest, or like what's been a question they've approached you with?
Yeah, so they've, it's interesting, it really is a bi-directional kind of like communication process. And so we actually, so for our BGP study that we're doing, actually our patient representative was very concerned that there would be a bias against smokers being offered BGP testing, because we know that kind of like, we know that like some of the mutations are less likely to be associated with smoking than for non-smokers, right? But from their perspective, that's inappropriate. And I can see that, right? So it's not ideal because there are claims-based methodologies for like detecting smoking using claims. They're highly specific, they're not very sensitive. So it's not ideal, but we're like, sure, we can do that, right? We can at least do a subgroup analysis where we try to see whether or not patients who we know are smokers based on this claims-based algorithm are receiving BGP differentially. So I don't know that we're going to be able to answer that one perfectly, but it was really important to them. They felt like that was something that was happening that was inappropriate. And then so that was something that and then we just really try to just keep the lines of communication open. We meet with them for each project. We meet with them at least twice a year. And then I'll put my CPC leader hat on, and we actually encourage all of our members to go, and we have a community advisory board, just like a generic one. So the kind of advisory panels that I was speaking about are all like we've developed from specific projects, but we have a community advisory board, and we encourage all our members to go and speak to them even before putting a proposal in, to make sure that their voice is represented in that proposal. And then to kind of follow up with them, or to create their own project-specific advisory boards as well. But yeah. Mm-hmm. Yeah.
Okay, go ahead.
It's
really quick. What data sets have you found have been the most useful for psychosocial data, like you mentioned depression?
Yeah, so again, we're using claims. So what we are going to be able to detect in the claims database regarding depression are going to be major depressive diagnosis, right? So it's going to be underdiagnosed. So we can look at things like diagnoses. We can also look at obviously we'll have to limit it to people with Part D, but we can look at people who are on certain prescriptions and things like that. Potentially, I think that that's less informative, but what I'm interested in terms of that being an outcome is like a major, like a new kind of major depressive event in the claims database. But yeah, that's an interesting question. And there, you know, that would just take me down the rabbit hole, which I'm happy to do with you sometime, about thinking through kind of all like whether or not that's something you would be more likely to find in EMR based data sets, right? And so there are some of those, they're, you know, like Flatiron data is more EMR based than obviously some of the, you know, obviously claims database is not. So thinking through like where we might be able to find some of that specific type of information, the different data sets that are available. So
Definitely needed, especially because depression is so prognostic for
Yes.
early mortality. I'm like, why is this not in every data set? But that's me as the psychologist, so thank you.
Totally related, the frailty,
yeah,
is that a,
claims based.
okay,
yeah.
so.
So there's
and it-
3 that have been validated.
Okay, oh, but like an algorithm, not
An
just.
algorithm, yeah,
Right,
yeah,
okay.
claims-based, there are 3 validated claims-based algorithms for frailty, one of which was developed at Yale. So we're gonna probably be doing looking across the 3 of them. Okay, great. Other questions? Oh, yeah,
One on the Zoom.
Yeah.
For your prediction model among 5-year survivors.
Yeah.
Are you able to apply your model in non-cancer patients to assess whether cancer patients have higher risk of non-oncologic outcomes compared to non-cancer patients?
yes, so yes, so in the, specifically in the Serum Medicare data, there is a cohort of non-cancer controls, so where we could do that. It's a different question and not something that we plan to do with this study. So using our like survivorship experts, they helped us kind of identify those 4 like non-oncologic outcomes. They help us identify both the oncologic and non-oncologic outcomes based on kind of like what was predominantly known in the literature. And so we started with kind of what the big ones were, right? But I think that there's a whole host of other things that we could look at as well.
Yeah, and for those non-oncologic causes of mortality, with the Serum Medicare data, will you just be looking at like comorbidities, other claims, or specifically at, like I know there's been research looking at cardiotoxic events related to like the use of ATAX or Taxate and
Yep, so we're looking at all.
looking at the chemo.
Yep.
Specific, okay.
Yeah, so like for specifically what you're referring to is like within the breast cancer
Yeah.
cohort, there's known kind of anthracyclines, things like that, which have been associated with well, I will say that the literature is actually mixed on it.
Yes,
So we are looking,
Okay.
and then also like radiation as well, right? So like side of radiation
Okay, yeah.
for breast cancer patients
Yeah.
specifically. So anything that we know, or at least there has been suggested in the literature is relevant, we're including if we're able to.
Last question.
No,
maybe,
you're
or
I'm happy to answer. Yeah.
You know, you did the study based on what your community advisory board suggested about people getting taken off medicines.
Yes.
Like, what was the context, like, did they feel like that was a lot or not a lot, or do we
I
have
think it
any
was less
kind
than
of?
they thought it was gonna be.
Okay.
You know, and it's interesting too, because it's, you know, trying to provide the context to lay people about like, also like, well, this is what we found, but this is why this may or may not be the complete answer to this question, right? So like, we found this, but this is in the Serum Medicare data, so these are the older, you know, the older patient population. Now, I would have expected, like, we would see the biggest concerns for toxicity in that population. And so, you know, I think it was less than they thought it was going to be. But, you know, 20% in 1 year, I don't know, like, I don't know what the right answer to it is, but just trying to be responsive to kind of their concerns as best as we can. Kind of like with the smoking, I don't know what we'll find, you know, and I don't know whether or not it'll make them feel better or worse about it, but we can at least be responsive to their concerns to a certain extent, so.
Another question, for your next steps for the RCC project, where
Yes.
you're looking at different data sources and using, because I saw that you had plans for EHR data specifically from Flatiron and CancerLink, was that
That's not for the RCC study,
Oh,
that's for the
sorry.
BGP study,
Okay,
yeah,
yeah.
yeah.
Is there a reason why you're using both? Is it just to like use as many different data sources as possible?
No, they have different types, so for
Okay.
both the Cancer Link, which is now the Concert AI data,
Yeah.
and as well as the Flatiron, they have different, neither of them is like what we
Yeah,
want.
perfect.
Right,
Yeah.
like, and so Flatiron specifically has the results from Foundation
Okay,
1 testing.
okay,
So we can get pretty good granularity for that specific
Yeah.
test,
okay,
whereas Cancer Link Concert AI,
yeah,
so I haven't yet gotten my hands dirty with the data, but from what they told us and what we understand in our preliminary analyses, they give you test results for tests that drove treatment decisions.
okay.
So there's gonna be an inherent bias
Okay.
introduced there, right?
Okay, yeah,
So we're not gonna just throw it out altogether and say
Yeah.
it's useless.
Yeah.
But we're gonna look into it, acknowledging that there's gonna be, so there will be some test results. And, they're saying that those test results then drove the treatment decision,
Okay.
but what that ultimately is gonna look like, I'm not sure.
okay,
So the Flatiron allows us to be a little bit more, I don't know, agnostic going into kind of like, oh, we
Okay.
just like look at but it's limited to like the specific test, right? Whereas
yeah.
we get probably more information across
Mhm.
multiple different tests in the Cancer Link
Yeah.
data, but there's
Okay.
some like selection that's happening
Yeah,
there,
yeah.
so.
Interesting, yeah, cause
Yeah.
we've actually used CancerLink before, but it's definitely where it's like, it's really great at some things, but there's like gaps in it. So
For sure.
that's why I thought it was interesting that you're using both of them, cause it's to kind of at least
Yeah,
give more information.
Mhm.
Yeah.
yeah, and you'll, as I described in all the rest of my projects, it's not necessarily trying to combine them, it's just saying, okay, like, these are the findings and how they're the same across these different data sets, how they're different, and what do we
Yeah.
think is driving some of these differences.
Kind of like a triangulation approach.
Yeah, exactly.
Figure out what you can learn from each
Yeah.
thing and where they're.
Mhm. So, and again, not perfect.
Yeah.
When people start collecting perfect data.
I know,
Yeah.
well, and that's funny, because people think like primary data collection is like the way around that, but I also have done primary data collection as part of like building our own cancer registries, and I can promise you that is also not perfect. Because you're limited by costs, you're limited by what you can get patients to fill out and like case report forms, like you're not gonna get everything, so
Yeah.
it's never gonna happen,
Yeah.
so I think.
It's truly this approach where you like look at whatever it is you have.
Yeah. Yeah,
It's really
matching, I teach a class called Health Outcomes Research, matching the right data to the right questions, where I try to instill this
Yeah.
into my students, like, you don't, you do it, you acknowledge limitations, you try to do it to the best of your ability, but you acknowledge that like you're never gonna be able to answer every aspect of the question.
Yeah.
In a single data set,
definitely.
I just have one question. I think value of information studies are so interesting, and I'm still trying to wrap my head around
So
those.
am I.
I'm curious, like, who's your intended audience for that? Who do you
Yeah.
want to hear the?
Yeah. So that's a great question. And that kind of gets more into the policy space. So what my PI, and I think is so interesting, and I started my career, by the way, I did a K99R00 and looking at Oncotype DX. So to some extent, this like just Oncotype DX, just in this year Medicare data, I was talking about like how you scope like an R1 versus a career development award. But it feels coming a little full circle for me, because there's still some parallels between what I did. But it's such an interesting question, right? Because to some extent, the horse is already out of the barn, right? Like, so we're going to do the value of information analysis. And so, yeah, I mean, there, some like of these like cancer societies will sometimes push for like kind of de-escalation of care when we find that you're not like the value of something isn't necessarily what we initially anticipated it to be. But with BGP testing, it's like they're already being widely utilized. We know that, even though no one's really looked at whether or not it even makes sense, or if it's even, like from a cost standpoint, whether or not it's really impacting. And so our study is going to be the first to kind of look at like, hey, like how often are we getting like actionable kind of results from these tests, right? And then does that translate to differential outcomes for these patients? And so the value of information piece is really like, to some extent, it's like, do we really expect to like, I don't know, like change practice overnight? Like maybe not, but at least like start asking the question is like, everyone's already using these. They're incredibly expensive, right? And so is it really kind of worth it? And do we need to kind of consider? Because I will say that we've had that grant for 3 or 4 months now, and one of the biggest contributions that neither one of us anticipated is just defining what BGP is, because like and it changes constantly because kind of everyone's coming with their new test, and like how do we want to define it and how are we going to look at it across these different data sets. There's the very obvious cases like Foundation 1 testing, Guardant, like things that we know, but then there's also like some institutions have their own panels, right? And then how do you detect that in these data sets with confidence? So that's been an interesting process. So
Thanks.
yeah. Okay. Well, thank you guys so much.